All right, the first thing that we are going to cover is population. So the idea behind this chapter is that now we are looking at natural populations. And so these are natural populations you would find in the wild. These can be human populations, for example. So there's over 6 billion humans on planet Earth. And we might want to analyze the entire human population altogether. Or we might want to analyze smaller populations. And so an example of that might be, for example, Georgia Tech students, or people that live in Georgia, or people that live in the United States. And we can do this for any species. We can do this for humans, but we also could do this, for example, for insects. So if we're interested in understanding the evolution of insects, or the natural variation of insect populations, maybe we restrict our analysis to all of the beetles in Georgia. And so to formalize a bit what exactly we mean by a population is typically what we mean is a group of individuals that belong to the same species. They tend to live in the same geographic area, and there is some sort of restriction in the gene flow, which means mating between different populations. And so this was certainly true, 500 years ago, 1,000 years ago. If you lived in different geographical parts of the world, there was a huge restriction on your ability to mate with those individuals. People tended to stay in one city or one region. There was little migration, and so the mating that took place was restricted to the individuals around you. Nowadays, of course, there's much more freedom of movement. We can fly across the world in less than 24 hours, and people move on a very regular basis. And so kind of this definition is being lifted a little bit for human populations. But the idea is that the population that you want to analyze, there's some sort of restriction in the gene flow between that population and other populations. And so the gene pool, now that we have a population, we're also interested in the gene pool of that population. So the alleles that are present in the population, the genetic information carried by members of the population, and the fact is that most populations contain high degree of heterozygosity. So there's a large number of alleles that are segregating in the heterozygous state. And so let's look at an example of this. So an example of this might be a pond, golden pond, or whatever you want to call your pond, and the frogs that live within that pond. And so if we take a look at the population that lives within this pond and we compare it to a population that lives in a pond across the country, there's obviously a lot of restriction in the gene flow between those frogs. Those frogs are very unlikely to ever mate with frogs that are found in a pond in California. And so each of those frogs is diploid, and so each of those frogs has its own unique set of DNA. And for a given gene, each frog has at least has two copies of each gene, and we might want to list for a given gene all of the alleles in the population. So we have here 16 different frogs, and each frog we have listed the genotype of the A gene. And so there's two different alleles that segregate within this population, and they can either be homozygous for the dominant allele, they could be homozygous for the recessive allele, or they could be heterozygate for each allele. And so we can take this set of 18 frogs and we can list the number of frogs that have this genotype. So there's 5 frogs within here that have this genotype. So here, let's erase this all. And we can list 1, 2, 3, 4, 5 frogs have the capital A. We can list the 1, 2, 3, 4, 5, 6, 7, 8. So there's 8 frogs that are heterozygote. And then there are 1, 2, 3 frogs that are homozygote recessive. So this is the genotype, and we can also calculate the genotype frequency. So this is the percentage of animals, or the frequency of animals that have the given phenotype. And so in order to get that, we just divide these number by the total number of frogs. So there's 18 total frogs. Oops, sorry, there's 16 total frogs. Let me correct that. So there are 16 total frogs. And so the genotype frequency is just a simple division of the number of animals that have each genotype. So 5/16 is the capital A genotype frequency. 1/2 is the heterozygote frequency, and then 3/16 is the homozygote recessive. And you'll notice that if you add these numbers up, they add up to 1. So that's one of the things, the genotype frequencies. The sum is equal to 1. We can also calculate the allele frequency. So this is the number of alleles that are present in the population. And so the total number of alleles is simply the number of individuals times 2. And that's because we're diploid, and so there's 2 times the number of individuals total alleles that we have to worry about. So in this case, there's 32 alleles that we have to worry about. And so simply we go and we count how many of each are in the population. So these 5 individuals have a total of 10 of the capital A allele because they each have 2. So 5 times 2 is 10. And then there are there's 8, not 5. There's 8 heterozygote individuals. And so there's a total of 18 of the capital A allele. And then for the little a allele, there's 8 here. And then there's 6 here because there's 2 times 3, so there's a total of 14. And so the allele frequency is another measure that we're interested in. And so that again is just you divide by the total number of alleles. This plus this equals 1. And the first number would be 18 30-seconds, and the second number would be 14 30-seconds. So these are given a name of P and Q. So in this case, P would be equal to 18 30-seconds. And so that's the frequency of the capital A allele. And then Q equals 14 over 32, and that's the frequency of the little a allele. So you notice that these numbers are completely separate from each other. They're not quite separate from each other, but in general, they're independent measures. You might want to think about this as, each frog rolls 2 dice. This ends up being a very confusing concept at first for students. So students very often confuse genotype frequency and allele frequency. Kind of one of the ways I think about it is like each frog is rolling 2 different dice. And those dice can be, between 1 through 6. So in this case, the frog has rolled a 1 and a 3. And so its genotype would be 1, 3. And you can imagine doing this for every single frog where they all roll dice. This frog is 1 and 2, etc., etc. And so the allele frequency is the number of 1s that were rolled in the population. The genotype frequency is you have to combine the 2 numbers together in order to get the genotype frequency. All right, so just to understand the different terms, the population, this is the group of individuals that are analyzed together. So here our population was these frogs. These aren't all of the frogs in the world. These are just 16 of the frogs. So our population is just a subset of these frogs. The population size are the number of individuals. So in this case, it was 16. The allele frequency is a measurement of how common a particular allele is found in the population. Expressed as a percentage. So P equals the frequency of one allele, Q is the frequency of the second, and then genotype frequency is a measure of how common a particular genotype is found in the population. So here the other thing I'll remind you of is that we just calculated this number for a single allele, so a single gene, but there's a large number of genes that exist in the population. And so two other things that are important to remember is that the frequency of each allele is mostly independent of each other. So let's say we are looking at 4 different loci in the genome, A, B, C, and D, and each one of those had 2 different alleles. So there is a big A and a little a, big B and a little b, big C and a little c, and a big D and a little d. So these are 4 different genes. So one thing to remember is that the frequency of these are all pretty much independent of each other. There is an exception if they are very close together, but if they're not close together, then you can consider the frequencies completely independent. And by close together, I mean on the order of 1,000 base pairs to 5,000 base pairs apart. And so the frequency of this allele is 0.8. The frequency of this allele is 0.3, the frequency of this allele is 0.24, and the frequency of this allele is 0.0014. So if we ordered which one is most common, that big A, the little b, the little c, and the little d are the most common alleles for this population. And so there's 4 different genes that we're looking at, and so each one has a unique P and a unique Q. And then, we can also look at different populations. So Walden Pond, frogs in Walden Pond, frogs in Golden Pond. And so if we calculated these numbers for these different ponds, very likely we would not have the exact same numbers. So, in one pond it's 80%, in the other pond it's 75%. In one pond for the B, the capital B, it's 30%, here it's 31%, and so on and so forth. So this is also true of humans, for example. So if I look at a allele in one population, in the US population, and I look at the allele frequency, and then I also calculate that in a completely different country. So if I calculate that in Canada or Mexico, the numbers are not going to be identical to each other. And so this is important, for example, for Ancestry or 23andMe or some of these other companies that allow you to infer your ancestry based upon your DNA. And so the reason that is possible is that all of these countries, this graph just shows you countries in Europe, all of these, but people from all of these countries have different allele frequencies. And so you can't do this analysis by just looking at one allele, but if you look at all of the alleles in the genome, or a large number of the alleles in the genome, then you can actually perform these statistical tests to kind of predict how much of each ancestry you have. And so this just shows you kind of one approach to take. So PCA analysis is kind of one of the ways that you condense thousands of different alleles, thousands of different genes together into something that you can visualize on a graph. And so these are just 2 components of that analysis. And so you can see, though, that individuals from different countries fall on different regions of the graph. And so this is what makes it possible to predict ancestry, kind of general ancestry, based upon your DNA.